Hi, everyone, and thank you for joining us. This is the last day of the Pura Syndrome virtual conference, and we are very excited to have a pretty amazing group of panelists who have joined us to help answer some final questions. That have emerged throughout the week. Some people have taken some time to submit questions in advance of this panel via our website. But for those of you who are with us today, you can submit questions. The most preferred method would be the Q&A, which you can find at the bottom of your Zoom screen. However, I'll also be monitoring the chat and we'll look for any questions there. But my name is Amanda Shanks. I'm the president of the Pura Syndrome Foundation. I want to give as much time to our panelists as possible for today, and then I'll be doing just a bit of a wrap up after that about the work of the foundation and what's on the horizon for us. And so I'm going to let each panelist introduce themselves to you for today. I'm going to start with I'm just going to go around the circle. And Dr. David Hunt, you're the first one on my screen. So would you just go ahead and take a few minutes to introduce yourself?
Sure, yeah. So I'm David Hunt. I'm one of the consultants in clinical genetics in Southampton, and I've been involved with Pura syndrome now since 2014. So at that time, we published one of the first two papers describing Pura syndrome. And since 2015, I've been fairly closely involved with the Pura Syndrome Foundation.
Thank you so much. He's been an incredible asset to us for sure. Dr. Matt Guile, I have you next. Oh, you're on mute, Matt.
I'm just unmuting. And my name is Matt Giel. I'm based actually not very far away from David in Portsmouth, just along the south coast. And it was through a connection with one of David's colleagues that my own group got interested in Pura just a couple of years ago. But what we do a lot of, we house the European Xenopus Resource Centre. Xenopus is a type of frog that's very heavily used in medical research and can be used nowadays to model genetic diseases. And so we've been making the Pura model for the last couple of years and trying to understand what happens very early in development when the Pura gene is inactivated. Thank you.
Great, thank you. Dr. Rob Rawl?
Well, yeah, thank you. I'm Rob Rowe. I'm a neurologist in Maastricht in the Netherlands. I'm not a pediatric neurologist, I have to say. I mostly see adult patients, but I see patients also in institutions for people with intellectual disabilities. And in this way, almost every genetic syndrome with epilepsy has my special attention. And Pura came to my attention about 4 years ago with my first boy with a Pura syndrome. That's how I got involved.
Wonderful, thank you. And Dr. Professor Dirk Niesing. I also said doctor, but it's Professor Dirk Niesing.
There's a doctor in between somewhere, that's fine.
Yeah, that's right.
So my name is Dirk Niesing. I'm a professor at the Ulm University in Germany, and we're running 2 labs at the Helmholtz Center in Munich and in Ulm. And we have been dealing most of the time actually with structural biology, biochemistry, and then actually Pura made us switching quite a bit our research, and we're now also involved in stem cell research and so on. I'm involved since 2016. I think David brought me in, and heavily involved, I would say, since then, and we're very happy to contribute and participate.
Wonderful. Thank you all so much for being here today. I'm very excited to have you all, and you represent such a diverse areas of both clinical research and scientific research. So this is wonderful to have you here to answer some of the questions that have come through. One of the first questions that came through via our website in preparation for this panel is a question regarding pain and pain tolerance. The question is, do Pura children feel pain? My younger daughter doesn't seem to feel pain at all. I've heard from others that as their children have gotten older, they start to feel it. Can you comment on this?
So perhaps I should start. So I think there are variable reports of pain thresholds in Pura syndrome. I don't think that there's a really consistent expectation of whether children with Pura syndrome will have increased pain thresholds. But certainly that is something that is of interest. And I know anecdotally there have been concerns about children who've been in pain and pain as a trigger for seizures. I think it's very difficult to measure pain in children who have impaired communication, but obviously there tend to be fairly overt outward signs that we look for in children as signs that they could be in pain. I think measuring changes over time is difficult, and perhaps Rob would have some thoughts about that as a pediatric neurologist, but certainly thinking about the Pura syndrome patients that we've encountered to date in terms of those that have been included in medical publications, we haven't really systematically inquired about perceived pain thresholds. And I think that that is something that is important, because we know that some conditions can alter pain thresholds. Objectively, I think that that is difficult to measure, but it would be helpful to have parental perspectives on that. And perhaps that's something that we could capture through the patient registry.
Yeah, I totally agree. I think there's 2 points to this. One of this, of course, is there's a subjectivity to pain and experiencing pain by the child itself, of course, but also by the parents. You can feel different about the pain which is perceived, and that makes it a very hard subject to really do research on, of course. And the second point is that in, well, almost every neurodevelopmental disorder, there could be any sensory abnormalities or sensory problems with vision, with hearing, and also with pain sensation. So it could be a problem, and I think we really have to look into that in the patient registry. Yeah.
Sure. It sounds like it's a very interesting area for us to look into a bit more. And some of what will help is gathering data and information from parents and caregivers through the registry, and then that might help guide, it sounds like, some further research into that. Is that how I'm understanding it?
Yeah, absolutely. I think that it's something that we could measure subjectively. So clearly we're going to be largely reliant on parental accounts, because there are obviously ethical issues regarding testing pain and patient responses. So that isn't something that we would be looking to do. But clearly, if there are clear reports of children who have experienced things which should obviously be painful and are not clearly distressed by that, then that would be an indicator that it's probably not a behavioral thing, that they aren't showing signs of distress, but that perhaps there are issues with pain perception. And I guess, you know, in some children they may have had things like nerve conduction studies, but those aren't great really for measuring pain perception. So for motor and sensory things, yes, you can get a gauge of how well peripheral nerves are functioning, but not those small fibers that are involved in pain. And I'm sure Rob would have more of an expert view on that.
Yeah, exactly. Well, these are just the nerves, and they do not say a lot about what a patient is really experiencing, what perception is. So it's just one, just one measure and one number which you get, but it's only, well, it's not even an indication of what pain perception would be.
It sounds like it's a bit of a challenge with patients being largely nonverbal, and that they can't necessarily communicate the degree of pain that they may be feeling. Any other thoughts on that from others? Okay. Another question that we received is, are there any structural differences you have noticed in the brains of Pura syndrome patients, either in the neurons and protein expression or in ion channels? If there are, what can be contributed to developmental versus a homeostatic role of Pura syndrome? Before you answer that question, will you please explain for those who are here with us today what that means?
So I think we had this question earlier in the week. So I guess the question primarily relates to the anatomy of the brain at a gross anatomical level. Are there any obvious structural abnormalities that are visible on imaging? And then there's a question about the histological appearance of the brain. So if you look at brain under or brain tissue under a microscope, can we see anything different about cellular architecture? And can anything be inferred about the function of neurons? So I think that that's probably what the question is getting at. And I guess most of the data that we have about the effect of Pura on brain development and function comes from animal models. But of course, we know from imaging that there are some common findings in children with Pura syndrome. So quite frequently, we find that there is delayed myelination during infancy and early childhood. There may also be some reduced brain volume and increased fluid spaces, and some one-off sort of structural changes that have been reported in the appearances of affected individuals' brains. We do know from the animal model of Pura syndrome that there seems to be a reduced neuron number in some parts of the brain, so in the cerebellum and hippocampus, and also the sort of cellular appearance under a microscope is different in terms of not just cell number, but also how the cells look, particularly the dendritic trees. So a lot of that work was done by Jennifer Gordon's lab. But in terms of looking at human brains, so there is one publication so far, which has come out of Alex Pachikowski's laboratory, in which they did do some very basic analysis of human brain tissue from a deceased patient and noticed some changes, but largely the sorts of things that we might expect in somebody with significant neurodevelopmental delay and a history of seizures.
Also, judging from the other side, when I have to diagnose a patient with epilepsy and intellectual disability, when we do an MRI scan, the findings in that MRI scan, the delayed myelination and some brain atrophy, we would call aspecific, so not very specific for Pura syndrome. So judging from that, the abnormalities caused by Pura in the brain have to lie on a deeper level than the gross structural level.
Thank you.
So I suppose what we're saying is that the appearances are relatively non-specific on a brain scan. There are some more specific findings from animal models, but in addition to the structural changes, we believe that there are important functional effects within the cells.
Absolutely. Okay. We did have a few comments come through related to the previous question on pain. One person put in the Q&A related to her daughter that she often spaces out with very unpleasant things that are done or painful things. One point when she was suffering from C. diff, she was virtually catatonic, is what is written. Any idea if this is found in other children?
So I have to confess that I'm not sure. So again, that would be something that it would be helpful to get more information about through the registry, because there will be, I'm sure, lots of accounts like this that haven't come to our attention. And again, they're not necessarily things that we would inquire about specifically. And I think it's really important that parents report these things through a natural history study, so that we can then think about specific inquiry and whether there seems to be whether this seems to be recurring in other children or similar events.
Mhm. Perfect. Just really
Would
emphasizes,
also,
it really emphasizes again that need to complete the registry once it fully launches. Just keep
yeah.
repeating that.
Yeah.
Yes.
What also would be interesting to know, because as a reaction to pain, it could be a blood pressure problem. That blood pressure drops as a reaction to pain. We call that a vasovagal syncope, and it might be that kind of a reaction. And there might be some hypersensitivity to get those reactions. That might be the underlying condition, and well, obviously you have to assess it and whether it is a problem. But whenever it's present, you could you could treat it with fluids to keep up to date with your fluid intake every day.
Mm. That's a really interesting thought on that as well. One person also put in response to the high pain tolerances that they met with a researcher in one of the meetups earlier in the week who's been studying pure beta, and that they brought up the smooth muscle and soft tissue disorders. And this particular caregiver is wondering if possibly all of the constipation, temperature regulation, for example, not sweating when extremely hot, super soft skin of Pura, and pain thresholds might all tie together from that standpoint.
It suggests that there's
Yes,
an autonomic component. Sorry, Rob.
Yeah, but we were saying the same, so no problem, David. Please proceed.
Yeah, so it sounds from those sorts of things that there could be involvement of the autonomic nervous system, and I think it's something that we've considered in the past. Again, although these things have been reported in a significant minority of patients, it's not something that would necessarily have been inquired about systematically, so it may be more common than we think it is.
I may add to this, because I think the person she was talking about is Sandra from my lab, and she's looking into pure beta. There's a study showing that pure alpha and pure beta work together to, express proteins that are important for muscle development. And we do have some doubts that the details of the study is true, and we're reassessing these things. It could be that this is all due to pure beta, but we're not sure about it. So, this is an open thing, but we have to disentangle those things. Yeah.
Sounds like it's a big lingering question at this point. Yep. But we're trying to find the answers. So, okay.
But I think the symptoms and the phenotypes David has referred to, they are there, and the question is just how is it caused, right? That's always that's the main gap we have to fill. The clinical view on one hand, and then what we see in terms of research, and we have to bring it together and match it, and that's where we're struggling.
Yeah, absolutely. Thank you. Another question that was submitted online is about a duplication. It says a family member was tested and the result says duplication on 5q31.2 with unknown clinical significance was the diagnosis that was provided. And the question is she at all in the Pura mutation? This caregiver struggles to understand what the exact address of Pura is. And it seems like her mutation is at the beginning or before Pura. Might be a very specific question, but anyone has thoughts on that?
I guess the key thing to establish is whether this is it sounds like this is a chromosomal duplication that possibly encompasses the whole of the Pura syndrome gene. Now, we're in the process of putting together some data that we'd like to publish, and Margot Rangers is leading on that, describing patients with chromosomal duplications that encompass the Pura gene. We think that there is a shared sort of clinical feature set between these patients, but it's completely distinct from Pura syndrome. So I guess the key distinction here is this a chromosomal duplication that includes the whole of the Pura gene, or is this a duplication within the Pura gene that actually disrupts the gene itself, in which case we would expect it to cause Pura syndrome. So that's the important thing that needs to be established. So I'd be very interested to hear from this family.
How can they get in touch with you, David?
You can get in touch with me through the patient registry. That's probably the easiest way. If you send an email to pura at sutton, SOTON.ac.uk, that would be the easiest way.
Any other thoughts on that particular question?
It's difficult at the moment. So the patient numbers are very low for duplications, and we're not entirely sure that the sort of shared clinical features are definitely due to the Pura gene, but we think that they probably are, because there are some patients with very small duplications that only include a few genes, including Pura, but most of the patients have larger duplications with very many genes. And so what you tend to do is you look at the features of all of those patients, compare their duplications, and you find the shortest region of overlap. And if all of the patients seem to have the same sort of presentation, then you look at the genes within that region of overlap and look to identify a candidate gene. And that's why we think that having too much of the Pura gene may cause a different disorder to Pura syndrome.
Absolutely. I think it will be very interesting when Margot's paper comes out on the duplications. I'm sure everybody will be, who's impacted by a duplication variant will be very interested to read those papers. So great. Thank you. Another question that was submitted online is, can you tell us more in layman's terms about the connection between Pura and Fragile X and ALS? How does this or could this relate to Pura syndrome?
Should I? David, do you want?
Happy for you to answer, Dirk.
Okay. So it's very different from Pura syndrome. In Pura syndrome, you have the Pura gene destroyed, disrupted, altered in a way that the protein doesn't function anymore correctly. Whereas the other diseases, they are, Pura is intact essentially. And what in these two other diseases happens is that, these are neurodegenerative diseases. So, the neural system gets worse and worse. And what people find is that there are clumps of things in the cell. And people have analyzed these pathological clumps of things in the cell, let's call them like that, and have found that there's, amongst other proteins, Pura inside. And they've been using animal models to find out then what role Pura might play, because one of the ideas is, well, if Pura is brought into these clumps, perhaps there's not enough Pura there anymore, to fulfill its normal function. And perhaps that's the cause of some of the symptoms of these diseases. It turns out in animal models, when you model this and have these clumps destroying the neurons, and you add additional amounts of Pura to it, the cells are healthy again. Now that sounds good, but this also works for other proteins, which makes it rather difficult. There's still a theory out there that Pura has a neuroprotective function. Pura helps to helps neurons to be stabilized, and to be less amenable to destroying or so and problems. And so that's something that could be a side effect if that is true. That when we find out how to deal with Pura and the Pura syndrome, and perhaps bring it back at some point in some way, that this could be, as a side effect, beneficial also for those diseases. But the cause is very different. Yeah. I don't know if David wants to add to this.
No, I think that's a good explanation. So clearly, the question that is of interest to the Pura syndrome community is whether the apparent involvement of the pure alpha protein in those conditions is actually important for the pathogenesis, so the sort of disease-causing effect in Fragile X or ataxia, tremor ataxia syndrome versus C9orf72, which is the frontotemporal dementia stroke ALS condition. And clearly we've been involved with researchers in the past who've reached out to us early on because they were interested in pure alpha, and we thought that it would be a mutually beneficial exercise, because it may be that that the involvement of the pure alpha protein in these sort of aggregates within cells in some way impairs pure alpha function, and that there could be a disease process in these patients which is in part due to the fact that pure alpha isn't functioning as it normally should do, which would suggest that pure alpha has an ongoing role within the cell, and that actually if you could then restore normal pure alpha function, you may modify the course of the disease. That would be the theory, but of course we don't know that that's necessarily the case. And it does raise an important question for Pura syndrome, because one of the key questions that parents often have is, if we could correct the Pura gene defect, would it cure the condition? And the answer is it would probably improve the condition, but it probably wouldn't represent a complete cure, even if you could change that in every cell of the body. And obviously we're mainly interested in the nervous system. But the reason for that is because obviously Pura has a really important role early on in the developing embryo and in brain development in general. And so if you were to correct that in adulthood, it would be somewhat like perhaps correcting the blueprints for a building after the building's been constructed. But actually, if part of the role is in terms of an ongoing process, and actually you just don't have enough manpower to undertake a certain function within that building, say there's a process and it doesn't have all of the workforce that it needs. If you could restore that workforce, then it may be that some of those processes normalize and that there could be some improvement. So I think it's multifaceted, but ultimately our understanding isn't so good at the moment that we would know for definite what the outcome would be. But I guess that the main interest in those conditions is whether we can learn anything about the role of pure alpha in those conditions, and does that relate to Pura syndrome in any way? I don't know if others have any other thoughts about that.
Thank you very much. One more question, sort of about, this is more of a clinical question, and then we have several questions related to possible treatments that families have submitted. This clinical question, though, is, does plagiocephaly affect brain and neuron functions, or is it just distortion of shape of skull?
Well, it's mostly this distortion of the shape of the skull. It might be that it's the other way around, that the disruption of the development of the brain may cause the different shape of the head, but mostly it's just distortion and positioning of the head in the crib or any other place where the child is.
Mm-hmm. David, do you have anything to add to that, or did Rob cover that?
No, I have nothing to add. I think of it in exactly the same way as Rob.
Okay, perfect. Thank you. So we, like I said, have several questions related to potential therapies or treatments that have been used for other rare diseases that people are interested in knowing just a little bit more about. The first question is, are there any therapies or drugs to reduce hypotonia?
Well, hypotonia is obviously an important problem in Pura syndrome. However, you cannot treat the hypotonia mostly itself. You can only treat consequences of hypotonia. So when children are very young, you have to treat the hyperventilation, you have to treat the feeding difficulties you encounter with the hypotonia, and you have to help give feeding aids. But there is no such thing, unfortunately, as a drug to alleviate that hypotonia. There are some case studies and case reports on later on helping children develop, and that might help a little bit. There is some evidence in other syndromes, in Down syndrome, for instance, in which children also can have a severe hypotonia, and therapy can help. Then treadmill exercise can help. Then obviously, all those children were already able to walk at that time. But in other genetic diseases, well, we can imagine that evidence is still very sparse, but you could elaborate on that, pulling the evidence from other diseases. That in that stimulation within the possibilities you have in your child is always good to see where improvement can be.
It sounds like one of the kind of greatest things that can be done to reduce, and it's not exactly reduction of hypotonia, but to address it, is physical therapy, occupational therapy, and speech therapy.
Exactly.
Okay. Great. Another question is, does the experimentation to synthetically create the pure alpha protein exist? Could this be a possible treatment or a cure?
I can respond to that because we're doing that in the lab every day, actually. So it's very easy to produce Pura. The problem is to bring it into the body and being stable, because all the proteins that are in the cells, they're destroyed, being made again, being destroyed, being made again. So even if we would be able to bring all Pura into all the cells, it would be gone after 2 or 3 days. So you have to bring in the information how to build Pura, the gene. That's the only chance you have to have a long-lasting effect, even if you were able to do that. So the short answer is, well, we can do that, we can produce it, and we can use it to study what the function of Pura is, but by no means we can have that as a therapeutic approach. We have to go through the building plan, the gene, if we want to bring it back something. And there had been the earlier question, I think, if one could at some point do that with CRISPR-Cas, and I think we were all very pessimistic that this will really happen, to do that on all the cells in the brain with the precision CRISPR-Cas has. For sure, you would end up with cancer. At least that's my personal opinion. But if we were to identify a very specific subset of cells in a region, perhaps in the brainstem, that is, really important and is also accessible by surgery or whatever, or microinjection, then perhaps at some point there would be a window of opportunity, because those things have been done in other diseases. But we're far away from that because we have no idea, which cells that could be. And if there are cells like that, perhaps it's a generalized function of Pura, then we don't have a chance, right?
Thank you, Dirk. It sounds like we have a lot more information that we need to collect, a lot more data, and it would have to be a very precision medicine type of thing in order to move in that direction.
And if I may add, I think one way to approach this is what Matt Goyle is doing, right? Using animal models, and he can look at where Pura is and where it plays a role during development, and see which parts it is important. And from that, we might at some point infer. I don't know, Matt, if you want to add something to that.
I think you're right, Dirk, that the current state of our knowledge is still a huge distance away from where we would need to be able to take advantage of any real advances in this form. And more likely, perhaps, is really being able to identify early sets of cells that are critically important, and then come up with other mechanisms rather than perhaps genetic ones. Because I noticed Jana's question there, talking about sort of gene replacement work. That has problems with it that David already alluded to, for example, where we don't know how early we would have to do this, even if we could do it, to have a really powerful effect on the disease. And also the question that we now see examples of people where they perhaps have too much Pura, and that is causing maybe not the same disease, but certainly it appears to be deleterious, albeit from early examples. So there is an enormous gap between where we are at the moment and where we would need to be able to take those sorts of very sophisticated approaches as they become available over the next decades, which they will, but it's decades probably before people get really good at these sorts of things. Even then, it may not happen.
Thank you. There's a question that just came in the Q&A. It said, we were recently told the most promising technique would be to turn up the volume on the working copy of the Pura gene. What are your thoughts on that?
I guess you come back to very much the same problem that we've been talking about. The first one is, if you turn it up too much, is that deleterious? And actually, my background is in gene regulation, and having really switchable ways of turning up gene activity in a way that you can really control is very difficult, even in a model organism, let alone in a patient or a variety of patients who have a lot of different problems.
Thank you. There have been a couple of questions that have come in regarding treatments and therapies that have been used for spinal muscular atrophy, for SMA. One is, how useful might a drug like, I'm going to say it wrong, Evrysdi, y'all can correct me, marketed by Genentech, and improving motor functions as it has helped in spinal muscular atrophy. And the other one is that the Zolgensma injection, having adeno-associated virus, which acts as a vector carrying the deficient gene to all cells in SMA patients, has been very successful for them. Is this a possible technique for Pura as well?
Perhaps I can start and then others join in. So this is a very different disorder. It's a disorder where neurons die. I think it's the second motor neuron that dies specifically. And it has turned out that this is somehow accessible, more accessible than things that are deep in the brain, because it's part of the spinal cord. And first drug was Spinraza that was applied to that, and were surprisingly really a specific gene activity could be switched. So the mechanism had been studied for 20 years. And so they could switch the gene activity. And now, we have these two additional drugs that you've been mentioning that make use of the fact that obviously it's approachable. And one is a small molecule drug that does the very same thing and alter the splicing, that's the word. But the other one is really gene therapy. That's true. It's, I think, an AAV-based approach, but it has the great advantage that these cells are accessible in a way, which for Pura will probably not. So I don't think it's of any use for it. But the question is, if one dares to try those things, that's a question I would refer to our MDs here.
So I agree with Dirk. I think the difficulty with virus-based gene therapies is always going to be delivery. And even though there are vectors that are effectively neurotropic or have good neurotropism and can be used for relatively good delivery, the problem is that we would want to restore Pura gene function in as many brain cells as possible, and even with intrathecal delivery, so injecting into the fluid space around the spine so that the agent is circulating in the cerebrospinal fluid, it still doesn't guarantee that you would have good levels of sort of transduction of cells. I don't know enough about the data from those studies, but my PhD was in gene therapy for the nervous system using herpes simplex virus, which is also a neurotropic virus, and sort of cellular sort of transduction, if you like, or transfection. That was always going to be the biggest challenge. And certainly with herpes virus, there was an issue of toxicity with the amount of virus that was needed, although I think that there have been considerable advances made with AAV, particularly I think AAV9, which seems to be the preferred subtype for nervous system delivery. But I have to confess that I've sort of been out of the game for a while, so I don't know what the current thoughts are really about virus-based gene therapy for the nervous system, and how widely applicable that technology would be, particularly if you're thinking about delivery and penetration of the brain as a whole.
Does anyone else have anything to add to that?
Perhaps the one note that those things are first tried with diseases that are lethal, where there's no other option, and certain death would come if you don't do anything about it. And so these first trials are usually a matter of desperation at some point. And I think with your own kids, I'm not sure if you really should do those things, right? Because it's untested. You have no idea what will happen. And your kids don't die at the age of 8, 10, 12. You don't see a slow, sudden death, which some families face, which is really, really, I think, terrifying. And you have a very different situation. So
we have one other similar question. Have any drugs to promote remyelination or prevent demyelination been tried on Pura patients? And an example was given, like some of the drugs that have been used for MS.
Yeah, that's an interesting question, of course. As far as I know, these medications have not been tried. However, most of those medications used for MS target the immune system at various levels. Well, the cause of the demyelination MS is really an immune response towards the myelin. So the myelin, she's the isolating part of the nervous system. And the demyelination there is mostly caused by that immune response. And when targeting that immune response, the demyelination is decreased, so you have better myelination. In Pura, the immune system is not that involved in the demyelination or in the myelination problem. So whereas you could try it, well, I obviously would not recommend it, but because, well, I guess the target mechanism is very different, very different.
It kind of sounds like there could be something interesting to look into there, but that it's maybe not something that you would strongly recommend trying in a clinical way at this time.
No, certainly not.
Okay. Matt, I'm just curious. I know yesterday in our closed GRN meeting, one of the things that we were discussing, there do seem to be a lot of questions about treatments, about drugs that might be used to help various things. And one of the areas we were talking about was eventually using some of our animal model studies to try out different therapeutic drug options. Do you mind just speaking a little bit to that? Because I think people would be interested to hear a bit more about that as well.
So two of the models that we use are two aquatic models. So the frog and the zebrafish are particularly well suited and broadly used for drug screens. Mouse are as well, but for very large drug screens, we can do these in plates that are kind of the size of your phone. And they will take 96 little fish larvae or tadpoles. And then we now have sophisticated ways of measuring the movements, and we will get more sophisticated at measuring different aspects of those animals. And of course, if you've got something, for example, I think the best example is tadpoles or zebrafish having seizures with the model, then you can try a whole range of really, you want drugs that are already approved for other things to see if they can have a positive effect. And that's your first screen, what people call a screen of compounds, to see if any of them could help. And this is something that might be, not too far away as an option as the models become more sophisticated and we're more sure of how well they're really mimicking aspects of Purus. So both of those aquatic models, my feeling is, I can speak firmly for the frog, less firmly for the fish, are still in their early stages, but are looking hopeful. And so that is an option in the relatively short to medium term, not very short. And as somebody who works in research, when I talk about short, I'm talking probably 2 years, 3 years, not tomorrow. But yes, that is an option. And as we get better at measuring things at high throughput in the models, then we will be able to do more things than just seizure-based movements. There will be other things that we can measure at high throughput that we could also screen for small molecules that might help alleviate.
Thank you. That's very helpful. It's also helpful just to get that clarification on short, what that means from a research perspective. Sometimes I think as parents, we think short and we're like, oh, next month sounds great. But sometimes these things can take months or years. So it's a great point. One question that just came in through the chat was, what about the drug Mistonin to improve strength?
I guess we're looking at the drug, which is an inhibitor of an enzyme in the synaptic cleft between the nerve ending and the muscle end plate. It inhibits the transmission of the neurotransmitter acetylcholine. It's used in a very specific disease we call myasthenia gravis. Also an autoimmune disease. And there you could use it in that specific disease to increase muscle strength and to alleviate symptoms of the myasthenia. As far as I know, it hasn't been used in other diseases, and when used in healthy persons, it doesn't increase your muscle strength anymore. However, I'm not completely up to date, but I think I wouldn't recommend it.
Is there any particular reason why?
It's a drug, so it has side effects. So because of its action on that acetylcholine level, it has side effects regarding salivation, swallowing, etc. And these can be very bothering for people. Also, people with myasthenia gravis can complain about those problems while they're still having good effects of it. And if effects are less clear than in that specific disease, well, I think the balance between side effects and effects are, well, I guess a little bit bad.
Sense. David, do you have anything to add to that, or?
No, I have nothing to add. Sorry.
Okay. Thank you. We have about 10 minutes left, and we do still have several questions that came in online ahead of time. I think this question was answered earlier in the week, but it's one of the most common questions that I know I see in our parent and caregiver group. So I think it's worth addressing again. And it is, will there be the possibility of applying the genome editing technique to the Pura syndrome in the future? Where has the Pura syndrome research kind of reached in that process, if it is a possibility? I know we talked about some aspects of that earlier, but I feel like if we could address that again, that would be great.
I can start. My lab does gene editing all the time on animals. it is not something that is easy to do at all yet in multicellular state. Once you get to many, many thousands of cells, unless you've got selection mechanisms and all sorts of things, all sorts of genetic tricks to do it. And it's, incredibly difficult, as David said from his own experience in his PhD, to deliver a gene modifying piece of DNA, and worse still, a complex with a protein, to very large numbers of cells. So it's a long way away. It also has the disadvantage of, at the moment, there are limits to the precision. It will definitely work where you expect it to work, pretty much, certainly in the models that we use it does, but you will also get some changes happen that you don't expect. And some of these are only really starting, as people do this more and more widely, to appear. So our lab and labs that we work with are finding new and interesting changes at sites that we're attacking, that we're not expecting when we're doing precision modeling, are happening more often than we had hoped. So, so I think the answer is, it's very difficult to rule anything out. We didn't have CRISPR-Cas in 2012. But at the same time, there's not a quick and easy route to using that sort of technology at the moment, by any means. I don't know if somebody wants to add to that, because I guess that there's people are thinking about some clinical aspects of CRISPR-Cas, but perhaps not in this case. I don't know.
Yeah, I would agree. I think it's very difficult to see how this technology would be used in vivo at the moment. So it may be possible for some conditions where, for example, if you wanted to take out bone marrow stem cells and to use the technology on those, and then to reintroduce them once you're satisfied with what you've done being correct and not causing any problems, then yes, that could be an application. But where we're thinking about using gene editing technology for the whole of the nervous system, the issue really is delivery and precision. And I think that those are two key issues that we will have to overcome before we really contemplate the use of the technology clinically. I may be wrong, there may be clinical trials that are underway, but to my knowledge, this technology isn't used therapeutically, licensed to be used therapeutically for any conditions as an in vivo technology at the moment. Does anybody know otherwise? No.
And if I may add, because my feeling is that the hopes are that if we were able to bring in Pura, the gene, and would correct that in all the cells, that this would resolve all issues. But remember, Pura is there from the very beginning. It's probably important before birth. So even if we were able to do that, it's unclear how much it would really be able to correct. There's wrong wiring of neurons perhaps, or too little. And I mean, the brain is really complex and can change over time. But the question is if you would get a full reversal, and I would be doubtful of that. And so perhaps a strategy that in the very end is, if we would compare a working gene therapy against something that treats the symptoms in an efficient way, perhaps the treating symptoms thing is the more efficient one in the end. I mean, we don't know, but it is not unlikely that this would be a better option, less risky, closer in terms of time. And then, yeah, of course, we have to understand what's going wrong on a molecular level, but I think this is perhaps equally good if we reach a point there. Yeah.
Thank you, guys. I have just a couple. These are a little bit more open-ended, and I would love to hear from each of you in response to them. I know we're not going to be able to get, there are a few lingering questions in the chat that I don't think we're going to be able to address on video, but we will see if we can gather some answers and communicate those out either by email. It may not come today, but maybe later next week, to any lingering questions that are out there. But I'm curious, just for each of you, if you wouldn't mind speaking a little bit to, and this is just your opinion, obviously, but if you could just speak to what potential research avenues are currently showing the most promise. I think that's a general question, but then there's also the most promise for a potential treatment. And then also, what do you think are the most significant gaps right now in Pura research?
Well, to start with the last, because that's the most easy to answer question, I guess, at least for me as a clinician, where I experience gaps, and also in when treating my patients and advise them on their antibiotic drug treatment, for instance, is what drugs can we use to well, to alleviate the epilepsy, which is often very refractory to treatment. So how to treat the epilepsy is a very, well, still an unanswered question, whereas we know that lots of patients with Pura syndrome have epilepsy. So that's, as far as I'm concerned, one of the major gaps I encounter here. But that depends on, and that's the most promising thing here, that depends on gathering lots of data to learn from each other, to see what treatment gave benefits to other patients with Pura. That's almost the only thing you can do as far as clinical research is concerned, on how to advance this.
David, do you want to go next? I think he might have frozen, actually.
He's frozen. Perhaps I jump in then.
Yeah, I agree with Rob. I think that actually from a clinical perspective, yes.
Oh, sorry, you froze for a second. Go ahead, David.
Yes, so I could see that everything was frozen on screen. So I agree with Rob. I think that from a clinical perspective, what would be really helpful is some evidence to support management. And again, this is the sort of thing that we would hope over time to be able to pull out of the patient registry, depending, of course, on the quality of the data that we're able to collect. But actually being able to give recommendations, evidence-based recommendations for management would be great, and has to be a key clinical goal. I guess in terms of the basic research, you know, our understanding really of the downstream effects of sort of Pura haploinsufficiency or intragenic mutations, which we presume cause a sort of functional sort of lack of protein within the cell. We really need to understand those things much better, because it may be that there are existing licensed drugs that could be repurposed that act on appropriate downstream processes and help to restore some of those functions within the cell. So until we better understand that, it's really hard to know where we go. But obviously we're doing lots of work across the research network to try to understand those things better, because I think we all recognize that until we do understand those processes, it's very difficult to think about where to go with therapies.
Absolutely. Thank you.
I think David just summed up exactly where we are. I don't know enough about the clinical to be able to comment, but in the research at the moment, would you say there's one thing that's particularly sending us in a direction? I will have a better understanding of this because I'm new to it, but I can't see something at the moment that where there's something that says, right, you must do this. It's the whole picture for the research at the moment at the cellular, developmental, biochemical, and genetic level. until we haven't got there, and to my mind yet. Dirk has a better understanding, so I'll leave him to sum up at the end. But it's not there yet. We need to look at the whole thing, and somewhere we'll find those clues, we hope. The sorts of clues that David said, those molecular clues that may tell us what pathways, what things that are happening within the cell, what networks are being affected. And then that might give us a clue as to things to try to alleviate some of the symptoms.
Great. Thank you, Matt.
Yeah, I fully agree. And I think what is lagging behind most is of course, clinical research, because the numbers are low. The numbers of patients are low. This makes it really difficult. Whereas the basic research doesn't depend on the number of patients, right? So we could establish move forward. And I think the establishment of the registry is a key step forward. That would give a great basis for a lot of subsequent work. And in terms of research, yeah, I think, already approved drugs, trying to find out if they could help, for instance, epilepsy. I think Matt alluded to that. That's obvious. And I think with at least most of the things we thought of to be important, we took care of, some of the components of the toolbox have been established, and we're collecting data. For instance, the stem cell work and so on, we have terabytes of data we're going through now and have to make sense out of it. But other stuff is, the tools have to be developed. Matt's lab and also the zebrafish, fish and frog, these tools are under development. So what is really missing is time, and you can't buy time. And then, yeah, I mean, in the short run, these are the things that make sense and we should go for. But finding out what goes wrong in terms of cellular events beyond epilepsy, this is much, much more difficult. And this takes time, and bringing up the spinal muscular atrophy again. There were 20 years of research to see that there are two sibling genes and one is broken. And if you treat the other copy in a certain way, it could mimic the function of the broken one. And that's how these drugs work, at least 2 of the 3 drugs that have been mentioned. But, this took 20 years of research. And I hope we're faster because technology has gotten so much better. But, finding, one calls it therapeutic endpoints. So things you can look at that if you improve them, would also likely help the patients. Identifying those points, that's what we have to do, and that's really challenging. And if we have that, we can also apply the drug screens to, to other symptoms that are more complicated. Yeah.
Great. Thank you. That's great to hear, both kind of a clinical perspective and that research perspective. Very helpful. This last question is a bit of a dream question for all of you as researchers, I imagine. But the question is, what would you do with a blank check toward making Pura research move faster and more efficiently? It's a great question. I'd love to hear the answer too.
I guess the easy answer would be, give everybody 2 or 3 PhD students. There's an upper limit of what a group leader can handle. And of course, things would get faster, but some things, can't be sped up, like producing, frog that lacks Pura. You have to have certain generations and so on. So money doesn't do the full trick. It helps a lot in certain aspects. But since it's unlikely that we would all get, funding of an entire lab, I think one has to set priorities. And I think registry, biobank, that's great. Administrative support, that also helps to speed up because, we also do that on the side. We all have teaching. Some of you see, most of the time patients. We have other projects, other research, and so on, exams we have to take. And things could be faster if there's at some point some positions that are bottlenecks. We would get some support, and then there are things that are hard to get funding for, which delays things. So if we have to apply for certain things that are hard to get funding for, and we have to wait for half a year or a year, and these are often not huge amounts, but the foundation could at some point jump in and say, well, this is something we provide money for. This would really speed up things. It's a little bit of putting oil here and there. I think that's a very efficient way of supporting us and speeding up. At least that's my take.
Thanks, Dirk. Anybody else?
Yeah, I agree with what Dirk has said. I think that money obviously oils the machine, but we have a fairly good idea of what we want to achieve through the research network, and key to that is definitely the registry linked to the biobank. We have people thinking about different animal models, about defining the molecular processes that are rendered abnormal when Pura is mutated, and ultimately we'll need to think about what the effect is cellularly and sort of at the gross anatomical level in developing organisms. But all of these things need to be understood, and I think that we're making inroads in all respects. But as Dirk says, having plenty of money would mean that yes, we would set up more projects in parallel with more PhD students or postdocs, but ultimately, it's really helpful to have things like administrators to support, as we've discussed with respect to the registry, because we're all fairly busy people trying to juggle lots of things. So I think actually having a lot more money wouldn't necessarily make things go exponentially faster. I think it would just having more money makes things easier because you can delegate activity to other people.
Thank you. I love that common theme, and it's something that the foundation continues to talk with all of our researchers about too, is how to kind of properly support and put more administrative support in place to ease the load on you all from that standpoint as well. I think there's going to be a lot of changes in the future with that. Rob and Matt, do you have anything you want to add to that question? It's a great question.
It's a very good question, and I can only really add to the fact that almost the only way, what we need is more time from people, whether that's people at the bench or whether it's David and Dirk and Rob having more thinking time on this problem. Then those are the valuable things. And in some ways, the thinking time is the most important time, because it takes quite a long time to do an experiment, whereas to have some time to talk and think around a problem can be relatively quick. And so really don't underestimate how effective putting in that sort of support in terms of administration to free that time up is. It's very easy to underestimate how it doesn't sound very exciting. It's not a big shiny machine. It's not an extra person in a white coat at a bench, but it's still extremely effective, because what it's doing is it's buying the thinking time, not only for us sat around here, but also for our senior colleagues who, we need time to think, and administration gets in the way.
Mm-hmm. I think all
Certainly,
of
yes.
Yes. Yeah. If you have additional hands to help do the work, it frees up that time to think. So, perfect. Rob, do you have anything to add to that?
No, these were already brilliant answers. I completely agree with those.
Okay, wonderful. Well, that is all that we are already over time just a little bit, and I know there are parties to get to and things to do this Saturday, and is my understanding from everyone. So I do thank you all very much for being here today and answering some of these final questions. For those who are in attendance, I know there are a few lingering questions still in the chat. If any of the researchers want to stick around for a second and type an answer to any of those, I would gladly welcome you to do so. If not, we will collect those and see if we can get some written answers and send them out via email sometime over the coming week. But thank you all very much for being here, for taking time over the course of this week and also today just to answer questions from our community. I very much appreciate each of you for taking the time to do it and for your continued work and research on Pura syndrome. So thank you all very much.